As usual, if you have any questions, kindly let me know in the chat. So today we're going to talk about Mycobacteria. I hope you can all hear me. So this, what we're going to describe is a general structure of the entire genus. For the specific ones, we'll mainly for Mycobacterium tuberculosis, we'll go through the epidemiology, the main pathogenic features and the symptoms. We'll also mention that for some of the other Mycobacterium species. However, there are too many that we can not talk about all of them, otherwise we'll be here the whole year. Then we'll discuss the lab diagnosis and treatment, again, with the main focus being on Mycobacterium tuberculosis because it's the most common one. And then we'll talk about the different, for the drug treatment, we'll talk about the different types of resistance that are known for TB, and then prevention and prevention and control. And then a few slides on Mycobacterium leprae and a few slides on tuberculous Mycobacterium infections. So as usual, we'll go through a classification, touch a bit on the general characteristics of the genus. And then for the specific pathogens, we'll talk about the pathogenesis, the clinical manifestations, microbiological diagnosis, susceptibility, prevention, and control. So the reason why I'm saying we can't discuss all of them is because there are more than 100 species of Mycobacteria. The main pathogens being Mycobacterium tuberculosis, that was first identified as the causative agent of TB in 1882. That's by Robert Koch, and is therefore alternatively known as the Koch bacillus or the tubercle bacillus. And again, more than 150 years down the line, we still don't have the full information about Mycobacterium tuberculosis. So the information about MTB keeps on changing, and therefore you have to read up on it to know what's current. Then we have Mycobacterium leprae, first discovered by Hansen in 1874, and therefore also known as the Hansen's bacillus. It's not as common as it used to be, but there's still spots in Kenya where it's found. And therefore, you never know where you land in clinical practice, so it's always good to know about things that may not be as common. So the rest of the microorganisms are found in the environment and collectively known as the Mycobacterium other than tuberculosis or non-tuberculous Mycobacterium or opportunistic Mycobacterium, and this cause opportunistic infection. So there must be factors that precipitate or predispose the patient to this particular species. In terms of classification, we have what is known as the Runyon grouping, which comprises the Mycobacterium tuberculosis complex that all cause TB, both in humans and in animals. And this list is actually increasing. So initially, we just used to have Mycobacterium tuberculosis and maybe M. bovis. That's the one that causes disease in animals, in cattle, but is also known to cause disease in humans. But then there's also the M. bovis BCG variant. That's BCG being the vaccine, the bacillus Calmette-Guerin strain of M. bovis that is what is used in the BCG vaccine, and that is also known to cause disease, particularly in children. And then we have different other strains. Africanum is more common in, I believe, West Africa and parts of Uganda. And then the different other strains that cause disease in animals, but are also known to cause disease in humans. Then we have the non-tuberculous mycobacteria, which include M. leprae and Mycobacterium lepromatosis, that cannot grow on culture. So they're not culturable, and they're not culturable, they're non-cultivable, so they do not grow in cultures at all. We have the slow-growing Mycobacterium that are non-chromogens, that do not produce any pigment on culture. We have the photochromogens, which are slow-growing, but produce either yellow or orange pigment when the colonies are exposed to light. And these include Cancer C and Marinum. We have those that produce pigment both in the presence or absence of light, that's scotochromogens. We have examples being Mycobacterium gordoni and Xenopi, and we have the rapid growers. So these take a very short time to grow. And these include the Chelonae fortuitum complex, Mycobacterium abscessus, and Mycobacterium smegmatis, which is found as part of the normal flora. In terms of general characteristics, they are all acid-fast, slim, bacillus-like or rod-like microorganisms. Acid-fast is based on the type of stain that is used, and we'll mention that shortly. They are all gram-positive like, so their structure resembles that of a gram-positive bacteria. However, they have some differences, and I'll show you that shortly. So they have the peptidoglycans, but in addition to that, more than 60% of the cell wall is comprised of lipids, and these lipids are what make it not easy to stain using the gram stain, and therefore we use a different kind of stain known as the Ziehl-Neelsen stain that I'll mention. However, even within that step, they resist decoloration by acid alcohol, and that is why they are known as as acid-fast staining. So there's a question as to why Mycobacterium leprae is non-culturable. It only grows, of course. I mean, I think they've either not been able to identify a culture media that they're able to isolate it on, but it also requires living tissue to be able to cause disease. And I believe that's why it's not been cultured. I hope that answers your question. They're all non-motile, they do not form spores. Some books say they're non-capsulated, but more recent studies have shown that there appears to be a capsule or capsule-like material around the cell wall, and therefore I'll not say it's absolutely not capsulated. There are more recent studies show that there's a substance or a structure that resembles a capsule. So please put this as plus or minus capsulated. And then they are all strict aerobes, so they can only grow in the presence of oxygen. They do not produce ectotoxins or endotoxins. There's one of them that we'll mention towards the end that causes disease through production of toxins, but it's not clear whether they're exo or endotoxins. Most of the diseases caused by mycobacteria tend to be due to response of the body's response to mycobacterium proteins, and this is what you've done in immunology that is known as a delayed type hypersensitivity reaction, and therefore some of them can actually be tested using delayed type hypersensitivity reaction for diagnosis. And we'll mention the specific ones. They all tend to cause a granulomatous response, and therefore most of the presentation appears to have a granuloma or granulomatous kind of presentation. And majority of them will cause a chronic disease, and therefore, from the time that the patient gets infected to the time you get clinical manifestation can run into years or decades. On culture, for those that are culturable, when you compare the human strains to those that, particularly in Mycobacterium tuberculosis complex, those that cause disease in humans and those that cause disease in animals, or particularly in cattle, we tend to show different levels of growth on culture. So when you culture this on a media that contains glycerol, the human ones will tend to have very heavy growth on culture, what is described as eugonic growth, while those in bovine strains will have very poor growth on glycerol-containing media. The cell wall is quite complex. I don't think it's been fully characterized to date, but as I said, it contains a lot of lipids, which take up about 60% of the cell wall weight, and most of it is actually what is known as glycolic acid. The other lipid components being the cod factor, which gives the bacteria sort of a beaded appearance, particularly post-treatment. And we also have wax-D that is used as an adjuvant in vaccines. So in contrast to what you see in the basic gram-positive bacteria, which would mainly be the peptidoglycan structure, the Mycobacteria cell wall tends to be much more complex. So we have polypeptides, an example of which is the PPD is purified protein derivative. And it's one of the antigens that's used either in our diagnosis of Mycobacterium tuberculosis, but also is part of what is used as a clinical diagnosis, where you inject what is known as the tuberculin test. And I think we'll show a bit of that in the diagnosis. As I said, we have free mycolic acids, but even the free mycolic acids have different components, including the free lipids, the glycolipids, and the peptidoglycolipids. And their structures are also quite different, comprising of different structures, and I'll not go into them. So we have those, we have the structure of the peptidoglycan that you know, the N-acyl, the NAC and the NAM, and we have the glutamic acids, diaminopimelic acid, and as well as different amino acids. And therefore, there are different structures, quite complex, and still particular high lipid content confers specific characteristics or properties to Mycobacteria. One of which I mentioned, that's impermeability to commonly used stains and dyes, and therefore we use the fact that it's acid fast for its diagnosis. Due to the high lipid content, most Mycobacteria tend to grow very quite slowly compared to the regular bacteria that take about 24 hours or less, so 16 to 18 hours. This can take sometimes up to weeks to months to actually grow on artificial culture media. They also confer antigenicity that I'll not go into details of. The colonies tend to clump, and again, I'll show a few examples of that. And then also confers resistance to many antibiotics. So we do not use most commonly used antimicrobial agents for mycobacterial species, they tend to have some selective kind of antibiotics and that can be used for this, and then also confers compounds, and for cannot, even most disinfectants are actually not able to kill mycobacteria. There's resistance to osmotic lysis, improvement deposition, and there's also resistance to most lethal oxidation that kill most within immune cells, such as the macrophages. So macrophages, I think, is below here, so you might not see it. This is they tend to form quite clumping colonies. Incidence rates from WHO 2021 report, so the rates in 2020. And so if anything that's wide, which I don't think is much of, is where there's no data. And here they say applicable, not applicable. I don't think it's clear what they mean, and they do not define what that means. But then if you move from the lighter portions, which is the Americas and some parts of the developed world, the lighter, what do we call this color? Turquoise color. Those are very low incidence areas. And as you move to the deeper green, then you have higher incidence. So most parts of Africa, and especially sub-Saharan Africa, will tend to have higher incidences of TB, as well as parts of Asia. And even within Africa, there are countries that have more than 500 per 100,000 population per year. In Kenya, I think we are at around 300 to 499, I believe, per 100,000 population per year. So the highest incidence rates are in sub-Saharan Africa and parts of Asia. Those are the countries that contribute the highest numbers of TB cases per year. So in 2020, there were 9.9 million cases reported globally. And that was the first year that I think there was an increase in the number of cases of TB in the last decade. This is probably not the most accurate in terms of identification of incidence cases, because we know COVID has probably been diagnosed. So the year before it was 10 million. So this 9.9, I don't think is the most accurate to WHO. There were 1.3 million deaths in HIV negatives and an additional 214,000 deaths in HIV positives. For the longest time, I think since 2017, 2019, TB has been the leading cause of death from a single infectious agent. However, I think COVID has overtaken that. And so currently it is the second leading cause of death, not from all causes, but from a single infectious agent. From all causes, we have probably heart diseases taking the lead. But when you look down, diarrheal diseases are also caused by infectious diseases, but we know that there are multiple agents causing this disease. So when you look down the column, I think TB is the only disease that's caused by purely one agent that is amongst these top agents or causes of death or disease. Now, if we then look at those countries that have more than 100,000 incident TB cases, they then come up with a ranking of countries by number of cases. And there are those that cause more than, or account for 2/3 of global cases. And this was in the year 2020. And those that are labeled are the ones that are ranked 1 to 8. So in Africa, 1 to 8, we have South Africa and Nigeria, majority of them being taken up by the Asian countries. But I mean, we are also in amongst the top producers of TB cases. So Africa probably has the largest number of countries in those top causes. But then when we talk about the top 8, then Asia is the one that contributes the most. Now, the fact that some countries, for example, countries like Canada do not have and do not appear in the top numbers, but if you then look, divide it by regions, you find that the parts of countries that do not show high prevalence of disease, but when you divide them into specific regions and look at specific distribution by race, then you'll find that they may actually then come up. But for now, let's stick with what WHO says. So 24th of March is the day, World TB Day, so later in this month. And this was first instituted in 1982. And this is basically commemorating the day that the Mycobacterium tuberculosis was first identified by Robert Koch. Various organizations, of course, involved in this. We have WHO, we have the Union of TB and Lung Diseases, and of course, we have the BCG 100 Day that celebrates 100 years of BCG. So there are many celebrations or days that, will I say celebrate, or that commemorate dates related to TB, whether it's TB Day itself or the day that celebrates the BCG. Okay, so in terms of specific, just to remind you of what the characteristics were. They are strict acid alcohol fast rods. They tend to be either straight or slightly curved bacilli. They're non-motile, non-sporing. They may be non-capsulated, but some strains are capsulated. They're strict aerobics. They grow at 35 to 37 degrees, but they're also facultative intracellular bacteria, so they can survive both within the bacteria, within the cell or outside the cell. Although for them to cause disease, they would need to multiply within the cell. They require specific media, so they do not easily grow on routinely used culture media in the lab, and therefore you will meet sort of new media when you do the practical for this. And what they require is enriched culture media. For a long time, this was used, the egg-based medium known as the Lowenstein-Jensen media. It's a solid media that contains, whose components are used for specific reasons. There's addition of malachite green in this media, so it tends to have a slightly greenish color, which is for inhibiting the growth of contaminants. So for this, we specifically need to inhibit the growth of contaminants because you're going to keep this culture for up to about 6 to 8 weeks before you declare it negative. So during that time, there's always the possibility of contaminations from either the environment or other, because you need to keep on checking the media. So you need to make sure that does not contaminate. And these are the other components within the media, and look up at what reactions are. We also have agar-based media or broth, and this is enriched with bovine serum albumin. This is used for different automated culture systems, where once you inoculate your specimen into the culture media, then the culture equipment is able to signal to you when there's growth. And that's either by production of detection of carbon dioxide or fluorescent systems. And then we have the liquid media, which so they tend to use the Middlebrook 7H10, 11, and 12. And depending on the culture program that you're using. Contributors to virulence of Mycobacterium tuberculosis include its ability to grow intracellularly, which protects it from the immune response. Its ability to detoxify oxygen radicals, basically preventing its killing. It has a slow generation time of about 15 to 20 hours, which that slow multiplication also protects it from the immune response. So it's able to hide either intracellularly or by multiplying very slowly. And then this we've mentioned, the high lipid content in the cell wall. And then there's the cord factor, and I'd like you to also read up on this. So this note, because it's a very long lecture, some of the points I'd like you to look up. Yes, too. And of course, if I meet you in the lab, I'll ask you. In terms of transmission, so you have an infected patient, and one of the most common presentation of patients with TB is coughing. And when that infected patient coughs up, particularly if they have cavitations in their lungs, which is one of the mechanisms by which it presents on X-ray that you'll be able to see once you get into clinicals. So that person who has a cavity within the lungs is able to cough up the bacteria. And if they cough up the bacteria, it gets, it forms what is known as an infectious or respiratory droplet. And these are very tiny collections of the microorganisms, of course, surrounded by what is in the air. And this can remain suspended for very long periods of time and can get inhaled by people who are within the vicinity. And that's one of the most common mechanism of transmission. So person to person by inhalation aerosols. And this is most common with Mycobacterium tuberculosis in people who have pulmonary tuberculosis, or TB laryngitis can also be transmitted by inhalation by droplets. For M. bovis, which causes intestinal tuberculosis, infected cattle produce milk that has the Mycobacterium, and therefore ingestion of unpasteurized milk or improperly treated milk or improperly handled milk can also transmit Mycobacterium bovis, which is part of the complex, the MTB complex. And then in the lab, through the skin, can cause contamination and can spread from there and cause tuberculosis, particularly extrapulmonary tuberculosis that we'll mention, and that's TB of the skin. Various factors, risk factors for TB, and this include, or this allow for progression from just exposure to established infection and to disease. For exposure, we have proximity to an infectious case, or duration of contact. We also have factors that play along with this, that's including overcrowding, poor ventilation, patients who take alcohol. This has been shown to affect the immune response, and therefore patients, if exposed to the microorganism, able to clear it before it establishes into infection. And of course, there's an occupational risk for people who either get contact through lab contamination or contact through what I wanted to say about this, but there's an occupational risk. And of course, that occupational risk is also healthcare, or people who work in the healthcare profession. And then for progression from infection to disease, and we'll mention this pathway, these other factors that come into play. We have the age, being the very young or the very old. We have gender. They say females are more, some individuals say females are more prone to progression to disease, which may be related to hormonal. But again, what has been found is that with WHO, I think they say the males tend to be the ones that present, or most cases reported tend to be males, and that may be due to financial aspects or socioeconomic status, where the males would probably go to hospital and the females not so much, because they're taking care of children or the home. And then we have the immune status coming into play alongside malnutrition and diabetes, have been shown to allow progression from established infection to disease. Overcrowding, again, poor ventilation, alcohol, smoking, and occupational risk also allow that progression to disease. So the very summarized portion of the immunology. Number one, I don't think it's still fully understood. Number two, very summarized because I do not want to totally confuse your life. So if we have this individual who has pulmonary tuberculosis, so they inhale aerosols that contain the Mycobacterium, and they bypass the basic immune responses that would clear them from accessing the lungs. So they get into the lungs. Within the lungs, they are recognized by macrophages. And what then happens is within the region where they settle in the lungs, there's then recruitment of other immune cells, including different other macrophages that are not infected, to the site. Monocytes, neutrophils, T lymphocytes, B lymphocytes, and dendritic cells. Once they come to this site, basically what they're trying to do is to clear out the microorganism. So what then happens is these cells then form or organize themselves into an area where you have the macrophage containing the microorganisms and other immune cells that have engulfed the microorganism, organize themselves in the center and are surrounded by other immune cells as well as fibrinogen and collagen. And this is what is referred to as a granuloma. So you then have the microorganisms surviving inside in the center part of that granuloma, and they are not causing disease at this stage. So this is known as a latent TB infection. And as this can remain as is in the human body within the lungs for an individual's lifetime. However, if that individual then gets immunocompromised for whatever reason, whether due to HIV, diabetes, malnutrition, the use of immunosuppressive agents, or any factor that allows the breakdown of this granuloma, then releases the dormant microorganisms. And once they are released, of course, they then become metabolically active and can actually then cause disease, and can then, if within the lungs, get exhaled or coughed up, and the cycle continues. I hope that simple explanation is clear. So that then gives you the different stages of TB that patients then present with. Okay, so exposure from an index case with open TB. Open TB is where they have a cavity, and therefore, or for any of them that are able to cough up the microorganism. And depending on the duration and intensity of exposure, as well as the ability of the immune defenses to clear the microorganism before it establishes infection, the patient or the individual doesn't get infected. However, based on this, again, there could be establishment of an infection. And again, based on the immune response, if the immune response is not able to control the infection at this point, or drive it into the formation of the granuloma, you then get uncontrolled bacterial growth leading to what is known as primary progressive TB. So the initial infection is known as the primary infection. And if the patient or the individual then directly progresses to active TB or disease where there's presence of symptoms, that is known as primary progressive TB. However, if that individual with a primary infection has a strong immune response that is able to control bacterial multiplication or bacterial growth, they can then develop or progress into the formation of the granuloma. Now, based on host bacterial or host factors or bacterial factors, the growth can get arrested at this point, and all the microorganisms can get eliminated. And that is what is described as sterilizing immunity. However, for most individuals, that growth gets arrested, but persistent of bacilli within the granuloma, known as the latent infection, latent TB infection, or LTBI. And that is where, kindly, there's someone who's not muted. If you don't mind, kindly mute yourself. Now, so majority of people who get infected, or about a larger portion of those who get infected, would then have latent TB infection. And this is estimated to be about a quarter of the global population has latent TB infection. Now, from here, we can then move into one of two scenarios. You can have the persistent immune response. So the individual remains immunocompetent, and therefore those dormant bacilli get eliminated over time, and that leads to clearance of the infection. However, for individuals who for any reason get immunocompromised, they then develop what is known as reactivation TB or secondary TB. I don't know why I can't see the bottom of this. Great, okay. So latent TB infection is the presence of Mycobacterium tuberculosis microorganisms, or the tubercle bacilli, without signs and symptoms, or X-ray or any form of radiological evidence or bacteriological evidence of TB disease. So these individuals are totally asymptomatic, and although they are not infectious, they do represent a group of individuals with risk of activation to active TB. So if they are treated, then that eliminates that progression to active TB. Now, the annual risk of developing active TB in these individuals is about 5 to 15%. And of course, that comes with any change in the immune response or their immune competence. And in HIV co-infected individual, this lifetime risk increases to about 50%. So, and this is not just with HIV. There's also increased risk with diabetes, with malnutrition, and other factors that have been known to act as risk factors for progression of disease to TB. So in terms of common symptoms of TB, most of them tend. Now, I think I'll stop sharing and start sharing because I cannot see the bottom of my slides. And I'm sorry about that. I still can't see that. Okay, so this prolonged cough, most patients that you will first have a high index of suspicion in are patients who have a cough of 2 or more weeks. Then they tend to have chest pain, but maybe when they're coughing, and sometimes even when they're just breathing in. Some patients will have blood or expectorate blood when they're coughing, or blood sputum. Sorry, this is not weakness in terms of weeks, but weakness and fatigue. They tend to lose their weight. So some of the patient will also tell you they've lost quite a bit of weight. They'll tend to have fever and chills, as well as night sweats. So usually there are guidelines as to how many of this you should look for. And that's why it's always important to take a good history when you're talking to the patient, find out what the symptoms are. We know that there are all these radiological and other diagnostic tests, but a good history can never be overemphasized because it guides you to what diagnostic tests to request. And that also saves on costs for the patient. However, it's not just in the lungs that Mycobacterium tuberculosis causes disease. There's also production or cause of disease in other sites outside the lungs. And this would occur due to, so when you have that rupturing of the granuloma, it can rupture either into just the surrounding lung tissue. It can rupture into a bronchiole, and if it does that, then it can spread to the same lung or to the opposite lung, which can cause, like if it develops, if it spreads into the 2 lungs or into 1 lung and spreads all over that particular lung, that is known as miliary TB, and I did that for clinical years. But it can also spread or burst into a blood vessel, and when it does that, then it can spread to various sites outside lungs, including the central nervous system, where it causes TB meningitis, into the lymphatics, and if it then settles and causes disease in the cervical lymph nodes, that is known as a scrofula, into the pleura, that's the covering of the lungs, and that is known as TB pleurisy. I mentioned miliary TB, where it gets disseminated into the lungs. but can also go to the bones and joints. That's known as Pott's disease, and into the genitourinary tract, where it causes urogenital tuberculosis. Extravascular, we'll get to TB diagnosis after this. So in terms of symptoms, they're varied. There are those that are specific, and there are also non-specific symptoms. We have productive cough, we have structural abnormalities, and that would be mainly when you take the X-ray. We have chest pain for TB, pleuritis, we have miliary TB. So there's some that are overlapping. So weight loss, so the ones that you specifically ask for would the cough, the fever, weakness, night sweats. Some patients will report poor appetites. And of course, depending on whether you're dealing with extrapulmonary, that will also, I know that you'd see a patient with persistent, and I know I've seen that where you see a patient with persistent back pain. You think it's just something happening to the spine, and you never even think of TB. You send for an X-ray and you get the characteristic diagnostic sign of a TB spine, and those are things that you just never forget. But let's move on with this. So this is a scrofula. So this is an enlarged cervical lymph node. You can have different presentation. This is also a case of TB. Any of the skin. This is spots that appears as that give us that abnormality on the spine can also cause that kind of spondylosis or twisting of the spine. And you can also have of the skin, and as we said, this can be either through inoculation through an abrasion, or mainly through abrasions, and is mainly from the lab. And those are the presentations of the skin presentation of TB. So in terms of lab diagnosis, specimens will vary. Sputum can be either coughed up sputum or induced sputum. Induced sputum is mainly done in children because they're not able to cough. So there's a way that you are able to induce them to produce sputum. You can also aspirate pleural fluid if the patient has a pleural effusion. Gastric washings are also commonly used in children because they tend to swallow the sputum instead of coughing it up. You can actually take or put in a nasogastric tube and aspirate the gastric washings and use that for diagnosis. Urine is also being looked at, and stool, mainly again for children. Aspirates of the different sites of infection, cerebrospinal fluid, fine needle aspirates, tissue biopsies, and the list goes on. I think it expands day by day. So in terms of sputum, you have to look at the quality. If the patient gives you saliva, then if it's unexpected, if they basically spit the saliva into a container, then that's not a good quality sputum. But it can be good quality if it's an induced sample. However, what we expect in most cases from expectorated samples is a mucoid-like looking sample. If it's bloody, it's also a good quality sample, or a purulent sample. So anything that looks like this is usually in the lab given a good, it's processed. Now, if a patient produces this, if it's induced, that means that in most cases, the doctor will be somewhere around. Then you need to indicate that it's an induced sputum. because if it comes to the lab and it's not indicated it's induced, then the lab does not process it, because to us it looks like saliva. So there's no use of really wasting 6 to 8 weeks waiting for results that are going to be negative. But if you indicate that it's induced, then it's going to be processed. So staining, we use a different profile of stains from what you know in microbiology. We use a Zell-Nielsen stain, and this I believe you should be able to see in the lab. And that gives you what are described as acid-fast bacilli, acid or alcohol-fast bacilli, depending on how the technique is done. The Quignon stain is actually a modification of this. And what you'd see from both of them would be the reddish bacilli, and that's characteristic. So this is usually due to one of the stains, the methylene blue that's used. But what you'd see is the reddish bacilli. They sort of change if the patient has been treated, where they tend to be bacilli, yes, but have some beading appearance. The other one we use is the fluorescent stain, which uses either Oramin or rhodamine. And this is what they look like. So this would be the greenish rods against, depending on what other stain you use, against the ones that we did show you would probably be against the greenish background. So the yellowish rods. This tends to be much easier to identify the microorganisms, even at low magnification. For the ZN stain, you need an oil immersion projection to be able to see. Our lab, I'd already mentioned that you use specific culture media. This is the gold standard in TB diagnosis. So you need to be able to isolate it to see for sure that this patient has TB. However, its advantage is that you require to incubate the media for about 6 to 8 weeks before you declare it negative. And I think I've mentioned this, so I'm not going to it. So solid media being Lowenstein-Jensen or Middlebrooks, the semi-automated liquid culture BACTEC 460 system. And we also have an automated liquid culture system that's known as the MIGIT, which stands for Mycobacteria Growth Indicator Tube culture system. Unfortunately, for latent TB, we do not have a gold standard, and therefore that's still in the air for identification. But I'll mention 2 methods by which we can identify potentially latent TB. So these are the culture media. So this is the Lowenstein-Jensen media with that coloration due to the malachite green. What does gold standard? Gold standard means what gives you a concrete identification of a pathogen in this case, but it can be gold standard for any type of disease. So in our case, to be PCR, I'll mention PCR shortly. To be able to say that definitely, so most of these other mechanisms, for example, PCR will tell me that I have genes or I have, so that the DNA for this microorganism is present. But it doesn't tell me whether this particular pathogen that I have, or that I have identified the DNA of, is still a replicating pathogen or is a dead microorganism. So for TB, the culture will be able to tell you, yes, you have Mycobacterium TB tuberculosis present, and it is replicating because it has to be able to replicate for it to grow in culture. While with the PCR, it does not tell me whether it's a replicating microorganism and therefore an active growing microorganism. I hope that explains it. So this LJ, this is the backtext system. So there's a way you have tubes in it, you enter you inoculate, and then it's able to tell you when you have growth based on either carbon dioxide levels rising to detectable levels or fluorescence for some of the other systems. Did I answer your question, Adnan? I hope I did. If I didn't, kindly let me know. So the colonies during electives, the Romanowski, James, some of these other stains tend to be used more in pathology, and so you mention them when you talk about TB in pathology, but not me. But there are, of course, other staining mechanisms. And for the gene expert, I'll mention that under PCR. So MTB colonies tend to be rough. As you can see from this, they're thick, they're wrinkled, have irregular margins. So their margins are not as regular as some of the other colonies that you see. And they are what is described as buff colored. And this is on the LJ or Lowenstein-Jensen media. Now remember that I mentioned that the Mycobacterium tuberculosis complex contains various other microorganisms apart from Mycobacteria tuberculosis itself. And some of those, for example, Mycobacterium carnetae tends to have smooth colonies. So they are different, definitely different from the Mycobacterium tuberculosis. And therefore, although they're classified as complex because they all cause TB in humans, they tend to have different cultural characteristics. And just in case somebody asks him. And therefore, to confirm MTB from culture, we use the growth rate. Takes quite long. The colonial morphology, the Zil-Nielsen stain will show almost similar characteristics of the acid fast for all Mycobacterium. So all Mycobacteria Zil-Nielsen stain show acid alcohol kind of features, acid fast, and therefore that's why you need other tests to identify. And then we have biochemical tests such as nitrate reductase test positive, the NSin test positive, and their catalyst test negative at 68 degrees centigrade. So this would be now specific to MTB. So other diagnostic tests include PCR, and PCR can be used from culture, but even more for some of them, you can actually use directly from a sample. In exam situation, are we supposed to know the different stains used for the different bacteria? Yes, you are. But then there's what somebody who is listed as Neisseria. So currently, what you're using for diagnosis, because it's much more rapid, is the nucleic acid amplification test, which is a PCR test. And what has been approved by WHO for diagnosis is what is known as the gene expert machine, which gives you results within 2 hours and basically identifies the Mycobacterium tuberculosis DNA and also at the same time resistance or susceptibility to rifampicin, which is one of the most important drugs for treatment of TB. But as I said, it doesn't tell you whether that DNA is from a microorganism that's replicating or not replicating. So it has its own limitations. So this is the test. You take your sputum, transfer 2 ml of whatever sample you're taking into a cartridge, place that cartridge into this machine, And within 2 hours, it has done all steps of PCR and has given you a printable result telling you whether the MTB is present and whether it's rifampicin resistant. So just a minute, I'll answer your question in a minute. So at least you're able to start treatment instead of waiting for 6 to 8 weeks, during which the patient is able to transmit infection to many people, and therefore, yeah. So there was a question here: what if you're misdiagnosed and given antibiotics? What impact does it have on the person clinically? Clinically, it depends. So if you have a latent TB infection, at least one of the drugs there that is recommended for treatment of latent TB will take care of that TB, of that latent infection. However, there's also the potential that if you have a low volume of microorganism, of course, there could be development of resistance. However, the only advantage of Mycobacterium tuberculosis is that its resistance does not develop as rapidly as for other microorganisms. It mutates very slowly and therefore does not cause as much resistance. However, if you have a person with resistant Mycobacterium tuberculosis, they can transmit that Mycobacterium resistant strain to other people. The other thing that would happen to you is you would be getting side effects of these drugs when you're not actually sick. So that would be a disadvantage. Yeah, that's probably all that I can think of at the moment. Why is it that despite most people being vaccinated at birth, we still have a quarter of the world's population having latent TB? Okay, so the current vaccination, I don't know whether I'm preempting discussions that I'll have later, but maybe I'll answer this question when I'm talking about control. And the last question, kindly explain the gene expert. Oh, you really want me to do that? So how, Kevin, maybe you could explain what you want me to explain about the gene expert. And if just to make it less complicated, there are actually videos on that show you the step-by-step how process runs that you can find on YouTube. If you Google the company name that is known as Cepheid, that's CEPHEID. If you Google that on YouTube, you can actually watch the video of how the test is actually carried out, including the mechanizations that are inside this machine that show you how it's pipetted and goes into a different chamber and all that. So I think at this stage, it will be totally confuse you, but you can get videos on that. If you would like me to send you the link, I can get you the link and send that. Is that okay, Kevin? Let me know if that's okay. I can send you the link. Can a patient who used TB drugs for 1 week transmit the disease? Yes. You're actually considered infectious until at least 2 weeks after the start of treatment. And Mary, could you remind me about your question on vaccination when I get to prevention? Okay. So immunological diagnostic tests, we have the tuberculin skin test, which has been used for a long time, where you inject what is known as PPD, that's what I described, the purified protein derivative, which is part of the cell wall, under subcutaneously, and then to form that bump or lump. And then the patient comes back after at 72 within 72 hours. So usually at the 72-hour mark, and what you're going to measure is there's going to be, depending on whether they are reactive or responsive or not, and this is basically dependent on the cellular mediated immune response, they'll form an induration, and that induration is what you measure, and then using the guidelines decide on whether they are positive tuberculosis skin tests, positive or negative. And those guidelines are dependent on whether patients have co-infections or comorbidities or other clinical conditions which may affect those results. So I'll not go into how many millimeters you're supposed to get to be positive. I'll leave that to the clinical because it's a complicated table, so I don't really want to go into it. Unfortunately, this does not distinguish between whether the patient has been vaccinated or has disease, and because individuals with advanced AIDS do not have a very good cell-mediated immune response, or patients with severe malnutrition, they tend to usually test negative. So it's not the best to fight. The other immunological tests are basically stimulation of the immune cells, targeting production of interferon gamma, and that is what the test detects. So it detects whether the patient is producing interferon gamma in response to Mycobacteria, is able to detect, to tell whether this patient has been previously or is currently having active TB, but again, does not differentiate from latent TB. So there's still a search for better tests because of this limitations of this immunological tests. I thought we'd take a break at this point. There's a question in the chat. Why is it? Oh, that's Mary's test question that I'll answer later. Okay, let's take a 5-minute break. Then I'll get back. So coffee break, water break, tea break, something break.
But akinini wewe mwenyewe tu unaweza nikwambia akiingia mara moja unamwambia tu ambie alien kama sisi tutamngaje mzazi
Okay. So I hope you're back. The blue lines were supposed to be my timers, but they backfired. So let's just continue. I'll answer your questions that are on the chat when I get to treatment and control. Okay, so in terms of treatment, the first lines have 5 drugs. So there's isoniazid, rifampicin, ethambutol, pyrazinamide. We use 4 of them. So previously we used to use a lot of streptomycin. I don't think it's used as much. Why? Because it required getting an injection of streptomycin every day for 30 days. So most people, I think, prefer using the other 4. So it's given as the 4 for a period of 2 months, and that is known as the initiation phase. And then we stop these 2 and we continue with the isoniazid and rifampicin for a further 4 months, and that gives a total of 6 months. So the second phase is known as the continuation phase. The option is rifampicin is for patients on heart or the HIV treatment. There's some drugs that interact with rifampicin, and therefore in that case you use rifampicin. The second line drugs, this is not a complete list. I think there have been additional drugs recently. That's paraminosalicylate acid, cycloserine, the quinolones, including ofloxacin, cipro, levofloxacin, moxifloxacin, I think is the more currently, one of the more currently studied drugs. And then we have amikacin, kanmycin, which are aminoglycosides, and capromycin and ethionamide. They've added in linezolid, and I know I have the other drug in my resistance profile, so that will come up. Question on treatment. What is the test of cure? A test of cure is basically, let's say, I'll use the name of the gentleman or lady who has called themselves Neisseria. Let's say you have a patient with Neisseria or with gonorrhea. So you've tested, confirmed that this patient has gonorrhea. You then treat the patient, then you rerun the test after treatment or whichever test you're going to use. Confirm that the patient has been cured. It's important to know which genes are amplified in mycobacteria during the PCR. For you at your level, probably not. I don't think anybody will ask you, at least in second year. You get, and that question I think I have not reached yet. So as somebody said, I found this interesting. So if you forget your TB drugs, you'll die, but you might need a priest. So to remember what your first lines potential drugs are, just use the name priest, pyrazinamide, rifampicin, isoniazid in hepatology. The drugs act at different levels. Most people at your level, again, at least in second year, might not ask at what level they act, but once you get into pharmacology and clinicals, they need you to know where they act. So aminoglycosides, we know inhibit protein synthesis. Pyrazinamide affects ATP, energy. Isoniazid, ethionamide inhibits synthesis of mycolic acid. Ethambutol, arabinogalactan synthesis. Cycloserine inhibit peptidoglycan cell synthesis. Inhibition of DNA by the fluoroquinolones. You already mentioned that, I believe. You already did your antimicrobial resistance. Rifampicin, RNA synthesis, paraaminosalicylic acid, I think was mentioned in terms of folic acid synthesis. So that's something that I found interesting. So various challenges in TB. We have individuals with comorbidities that put them at risk of TB, and this for the longest time, I think the most studied has been HIV. But then we have others such as diabetes, patients with systemic lupus, erythematosus, patients, and more recently, I think even individuals, helminths have been found maybe to have in susceptibility to TB. Patients with particularly HIV have been shown to have atypical presentation, as well as bacteriological presentations. So experts fortunately is able to tell us about rifampicin resistance. We also have other tests known as line probe assays that tell us about resistance to both first line and second line drugs. So drug resistance can either be monoresistance, which is resistance to one drug, any of the drugs. It can be polyresistance, which is resistance to more than one drug. And when it comes to the polyresistance, we then talk about multi-drug resistant TB. Now, this is defined as tuberculosis that is caused by Mycobacterium tuberculosis strains that are resistant to at least these two drugs. So it can be resistant to any of the other drugs. As long as these two are in that group of drugs, then it's known as MDR TB. Then we have extensively drug resistant TB, first diagnosed, I think, in South Africa. And this is TB, which alongside being an MDR TB is also resistant to a fluoroquinolone and at least one of the three injectable second line drugs. So amikacin, kanamycin, and capreomycin. So this was recently redefined to MDR TB strains, so resistant to rifampicin and isoniazid, but also resistant to a fluoroquinolone and either linezolid and bedaquiline. That's the drug I was trying to remember. So this is, I think, as at the year 2020. 2020 is when it was revised. In terms of TB drug pipelines, we know the different stages of development of a drug. There's several drugs that are being looked at different stages, and I think the ones that are closer to the end include delanmanid, gatifloxacin, moxifloxacin, rifampicin, and I think there have also been a few studies on bedaquiline and linezolid. So in phase 1, I think currently there's nothing, but still quite a number of agents being looked at. So here we come to the BCG vaccine. So prevention and care, I think BCG vaccine has been used for the longest time, and it's given at birth or as close to birth as possible. There was a question as to why the tuberculin test. I'll repeat that, just give me a short one. There was a question as to why is it that despite most of the population being vaccinated, I think by BCG, using BCG, that a quarter of the world still has latent TB? So the BCG vaccine, unfortunately, has very variable protective or protection, and it ranges from as low as 5% to about 80%. And most of the protection it gives seems to be in young children. and it seems to wane over the years, such that in adulthood, there's minimal protection. And even for children, what has been found is that it seems to protect them from severe infection, severe being particularly the complications of TB infection. So the more, for example, TB meningitis or severe chest pulmonary TB or more severe infections, not so much the more the initial infection. So that is why there has been a hunt for more appropriate or more effective vaccines, and that's still in the pipeline. In terms of prevention and control, respiratory isolation, so patients are supposed to be isolated for up to 2 weeks until they are considered non-infectious. We used to keep that at 2 weeks. I'm not sure what the current one is and whether people are able to isolate with the large numbers of people admitted. And then we have ventilation of households, contact tracing. Nutrition is important, of course, because of it works in collaboration with the drugs. And then chemoprophylaxis. Vitamin D has been thought to have a role to play. That is still under investigation. Okay, wow. I think I might defer the questions to the end because otherwise we'll never finish. There was a question on the repeating the tuberculin test. The tuberculin test uses a component of the MTB alcohol, MTB related or associated antigenic proteins. So we use PPD, that's the purified protein derivatives that's found in the cell wall. It's part of the cell wall, a component of the cell wall. So it's injected subcutaneously into the skin, and then the patient goes out. So when you inject it, you'll get that small lump that's forming, and you draw. So you'll have that because it's a solution that you're injecting. So you'll have that small lump that forms. So I would draw or mark around that area with a pen and ask the patient not to rub it off or wash it off. And then the patient goes home and they come back around the 72, usually should not go beyond the 72 hour mark. And what you'll find is an induration or it's a skin reaction. I'll try and get a picture of that. I thought I had one. And what you would then measure, and that is, sorry, so you would measure the skin induration that will form around that area if the patient has a cell-mediated immune response to what you've injected there. So you'll get, it usually looks like a slight elevation of the skin around, like a swelling, like an allergic, similar to an allergic reaction. It's not an allergic reaction, but you get similar to an allergic reaction. And then you measure that, and using the guidelines, you then define whether that is a positive response or negative response, which is also determined on what other comorbidities or co-infections or clinical conditions that that patient has at the same time. Is that better? Please read about it. It's maybe something that you may use quite a bit. I will skip this. Okay. Again, because I'm sure you have a class. And these are some of the vaccines that are currently at different stages of trials, including a BCG revaccination. I think is also one of the ones that's being tried. So where they give you a booster of a BCG vaccine and see what the immune response is. So this again have at different stages of trials. Now let's see whether I can finish this in the next less than 20 minutes and then answer the rest of your questions. So Mycobacterium leprae is an obligate intracellular organism. So it does not grow outside cells and has been shown, apart from the human cells, it has been shown to grow in the pad or in the foot of the mouse or in an animal that is known as the armadillo, which is this particular microorganism, and the mouse is here. The reservoir is infected humans, however, it shows very low infectivity, and that's why you don't hear so much about it. Transmission is mainly skin-to-skin contact, and it tends to grow best in the cooler part of the body, including the skin, of the eye, and the upper respiratory tract and the testis. The incubation period ranges from about 3 to 15 years. It's a chronic lepromatous disease. I've said it involves the skin, peripheral nerves, but also the mesomucosa. And transmission, I've already mentioned that. The main target cell of the bacilli is the Schwann cell, which then results in nerve damage. And this nerve damage is what causes the characteristic features that include anesthesia. So there's no sensation around that area, as well as paralysis of the muscles. And the first sign, however, is a non-specific sign and looks almost similar to a ringworm, if you've ever seen a ringworm. It's classified depending on the host immune response into two classes. We have the tuberculoid leprosy, or the tuberculoid type, which has the tuberculoid leprosy and the borderline tuberculoid leprosy. These tend to have very good or relatively good cellular mediated immune response. Or the lepromatous type, which has three groups: the mid-borderline, the borderline lepromatous, and lepromatous leprosy, which has a very poor cell-mediated immune response, but very good antibody-mediated response. So from the tuberculoid leprosy, the cell CMI reduces as you go towards lepromatous, while the antibody reduces as you go towards the tuberculoid form. So based on that, based on the CMI, or the cell-mediated immune response, you have very few or no bacilli in the tuberculoid leprosy, and therefore it has very few clinical lesions. So you'll get either one or just a few hypopigmented macules or plaques that tend to be very well demarcated and hyperesthetic. The patient tends to be devoid of normal skin organs, including the sweat glands, and they do not have hair follicles, and their skin tends to be dry and scaly. The nerves may be damaged with loss of sensation, and they do not sweat, what is known as the others phenomenon. And when they then get injected with bacilli that have been deactivated or boiled, what is known as the lepromine test, they get injected with that, they tend to be positive because they have a very good cell-mediated immune response. And therefore, these are the lesions. So they just look like what is known as mashlingis. You can actually tell where the border is. And from the middle, you sort of see a change as you get towards, it seems almost like it's healing from the middle as you get towards the edges. Some similar presentation. Well, lepromatous, in contrast, because they have a poor cell-mediated immune response, they tend to have very large lesions with large number of bacilli, even where the skin appears normal. They have symmetrically distributed skin nodules, so on both sides of the body, with poorly defined margins. The nerve damage is so severe that sometimes, even when they get their digits or their toes and fingers get cut, they actually do not feel it. The late manifestation include loss of their eyebrows and eyelashes, dry scaly skin that's particularly found on the feet. And in this case, because the lepromine test is a response to, is an indication of the cell-mediated immune response, similar to that found in TB, the lepromine test is negative. So they're just some of the lesions. So they get, because of either the anesthesia, they do not feel their nerves due to the nerve damage. They can get a lot of ulcers, their bones also get resorbed and also get cuts, and then you have all these foldings, very minimal eyebrows, eyelashes, and folding, sort of like corrugated kind of skin lesions. In terms of diagnosis, remember this microorganism is not cultivable in the lab, so we do not culture because we do not get anything from cultures. Specimens include nasal discharge and scrapings from the nasal mucosa, as well as slit skin smears, which I think you do more of in pathology. Microscopy, we use the ZN stain again, and we get acid-fast bacilli that tend to be together accumulated intracellularly into masses, what are known as leprosy globi, and this is mainly lepromatous leprosy. This is better seen with a modified ZN stain known as the fight stain. And with a slit skin smear, you can usually determine the bacterial index using the fight stain. You can also use the fight stain for punch biopsies of skin. And of course, you can always use PCR for everything. Treatment include rifampin, dapsone, and clofazimine, and is prolonged, takes sometimes up to 24 months before you start seeing a reduction in the bacterial load. Second line include ofloxacin and minocycline, but you can also use triple drug combination in cases where a patient has only a single lesion. That takes longer. And you can see it actually clears, so the patients can actually get healed, especially if you catch them before they get those damages of their digits. In terms of prevention or control, detection and treatment of infectious cases, early detection of diseases, particularly in those close contacts. And for children that are exposed, you can use dapsone for chemoprophylaxis. And it's been shown to have, BCG has been shown to have some varying results in prevention of leprosy. And maybe that's why, maybe it has better results than TB. That might explain why we don't see much of it also. But again, not clear whether it's BCG or the fact that it's only found in certain locations. I'm not sure exactly why. Then let's finalize with the NTMs. So the non-tuberculous mycobacteria. Again, quite a number of species. We already mentioned their synonyms. So it's MOTS, not MOS, it's MOTT. A typical mycobacteria, environmental opportunistic. They show variable geographical distribution globally, and as well as variable pathogenicity. And in most cases, they tend to be found in immunocompromised hosts. So there's Mycobacterium avium and Mycobacterium intracellulare that have been shown to appear as a complex because they're very difficult to differentiate, tend to be found in soil and water, and therefore transmission is mainly through inhalation and ingestion. They've been found to cause disseminated infection mainly in immunocompromised hosts, and have been actually found to be quite prominent in HIV patients, and present with pulmonary disease patterns that are very difficult to differentiate from TB, and therefore can only be identified using diagnostic tests. This pathogen tends to be resistant to most antibiotics, and to most anti-TB drugs. So it's always important to identify what they are, what you're dealing with exactly. They tend to be fairly susceptible to macrolides, such as clarithromycin, azithromycin, to rifamycins, rifampicin, and rifabutin, and to ethambutol and a group of other drugs. But the first line is mainly clarithromycin, rifabutin, and ethambutol. Unfortunately, if you're using the gene expert, I think it doesn't identify the MAC complex. So that becomes a problem because the clinical presentation is the same as TB, but it's resistant to most TB drugs. So you'll not be able to use a 4-drug combination in this case. Your patient will not improve on treatment. Then we have the Mycobacterium fortuitum chelonae complex, which has been found to cause abscesses in patients who inject themselves, or post-traumatic abscesses, but also post-surgery in patients who have heart valves, mainly through contamination. Remember, they're found in the environment, or causing corneal infections in patients who get eye injuries. And treatment for this is amikacin, doxycycline, as well as surgical excision, particularly in abscesses. If it's in a prosthesis, you may need to remove it. So, and then we mentioned scrofula, which is cervical lymphadenitis. Based on age, the sources are usually different. I mean, the causes are usually different. So in most adults, you'll find it's due to Mycobacterium tuberculosis. In children, you'll find it's due to mainly Mycobacterium scrofulaceum, which is found in the environment and presents as a chronic painless mass in the neck, also known as a cold abscess. So you will treat based on what the positive agent is. Scrofulaceum will involve surgery because it's usually resistant to antibiotics. And if it's due to MTB, then use anti-tuberculosis. And I think I've shown something similar to this, and that cervical lymphadenitis. And second to final, we have Mycobacterium marinum, which is also known as the fish tank granuloma or swimming pool granuloma, usually found in people who clean fish tanks. People get in through scratches and abrasions, and is similar to what is seen in cutaneous tuberculosis. And usually they tend to be localized, but they also form a tracking kind of dermal lesions where they track the lymphatics, and this is known as sporotrichoid, if I can just show you. So something, it can be either local, or it can sort of track the lymphatics, which is similar to something you'll see in one of the microorganisms you'll go through, one of the fungal agents known as Sporothrix schenckii, and therefore it's known as a sporotrichoid spread. It tends to be self-limiting, but you can also use antibiotics such as minocycline, cotrimoxazole, or rifampicin with ethambutol. And finally, Mycobacterium ulcerans, which causes Buruli ulcer, I think first identified in, should be Uganda, in low-lying marshy areas that tend to have periodic flooding. And it causes ulcers that, I can show you this first, that start off as a papular nodule, then becomes a plaque, then edema forms around the area, and then it ulcerates. They can be relatively small or quite enlarged, even much larger than this. And so depending on the stage they are at, treatment varies. In the early preulcerative lesions, you can use excision with primary closure. For those that have large ulcerated lesions, you can excise and carry out grafting, and antimicrobial agents tend to have variable results. Yeah, so they can be as big as that. Now to the questions that I have not answered. Okay, how far back do I go? I repeated that, answered that. If an exam, if I or someone else asks you which two drugs must MDR be resistant to, do we choose rifampicin and isoniazid or bedaquiline and linezolid? So MDR is specific to rifampicin and isoniazid. The additional bedaquiline and linezolid was for XDR TB. So MDR has to be at least resistant to rifampicin and isoniazid. I hope I answered your question, Abdallah. And the clinical explanation of night sweats, I honestly cannot remember, but I'm sure you will know about it before you get to clinical yet. It automatically, like when you hear of night sweats, automatically clicks to TB, not necessarily, but it's one of the features that they'll actually mention. Yeah, I honestly cannot remember. I had a slide that actually explained this, but I thought it was too complex, so I removed it. But I could answer that question on, if you don't see me on Friday, then please, the next time you meet me, even on the corridors, just ask me. Is it a must for one to have a scar in the place where the BCG was injected? No. Some people do not develop the scar because of maybe the level of the cell-mediated immune response that they get. And depending on where you are, some countries will insist that you get the BCG again if you're coming from TB endemic areas. Yeah, but not everybody gets a scar when they get injected. And then based on the BCG scars, you can actually tell which country people come from. I think there's some country in Africa that give it on the right side, the right hand. As in Kenya, I think we get it on the left. Thank you very much. Anyone with questions? More questions? What is fungal? The fungal infection, did I say similar to, let me send you the microorganism because I think that was the spelling. I may have gotten it wrong, but that's the microorganism that causes the sporotrichoid presentation. If you have any more questions, I'll hang around for about 5 minutes.